Okay, let's get right into it. Today we're tackling a really fascinating and, let's be honest, a truly challenging bacterium, Elizabethkingia meningoseptica. This is an organism that seems to delight in breaking all the rules we expect to see, both in the lab and at the patient's bedside.
So, let's kick things off with a question that gets right to the heart of the paradox. Which gram-negative rod is just naturally, intrinsically resistant to our heavy hitters? Our go-to drugs like Carbapenems and Colistin. Yep, if Elizabethkingia popped into your head, you are absolutely on the right track, and that's exactly the puzzle we're going to piece together today.
Now, before we really dive deep, just a quick note for you. All the information in this explainer comes from a Gemini deep search. And for those of you who really want to get into the weeds, we've made the full list of sources available in the linked notebook document.
So here's our game plan. We're going to follow the entire diagnostic journey, almost as if we're walking in a microbiologist's shoes. We'll start with the clinical picture of this sneaky thread, then move to the lab bench to see the first clues. From there, we'll unravel its deceptive nature, wrestle with the huge therapeutic dilemma it presents, and finally, we'll talk about how on earth we can control it in the hospital.
All right, first stop on our journey, the clinical picture. Elizabethkingia meningoseptica is a really serious nosocomial pathogen. But here's the thing, it doesn't just infect anyone. It's a true opportunist, and it picks its targets from among the most vulnerable patients we have.
And the diseases it causes are incredibly severe. It's probably most famous, or infamous, for causing these absolutely devastating neonatal meningitis outbreaks, often in ICUs. In adults, particularly on the ICU, you're looking at life-threatening bacteremia, frequently linked to central lines, or ventilator-associated pneumonia. It is the definition of a hospital-acquired threat.
And the stakes here are just unbelievably high. In some of these neonatal meningitis outbreaks, the reported mortality rate has soared as high as 57%. Think about that. And for the babies who do survive, the fight is far from over. Many are left with severe, permanent neurological damage like hydrocephalus. It's just heartbreaking.
Okay, now let's pivot from the bedside and head over to the lab bench. When this organism finally gets isolated, what are those first initial clues that a microbiologist sees?
Well, you put it under the microscope, and at first glance, it looks pretty standard. You'll see these slender, maybe slightly curved gram-negative rods. It's non-motile, doesn't form spores. So far, nothing's really screaming for attention.
But here, this is where things get interesting. It grows perfectly fine on blood and chocolate agar. You get these nice, smooth, grayish colonies. But, and this is a huge but, it shows poor to absolutely no growth on MacConkey agar. For a gram-negative rod, that is a massive red flag. It's the kind of thing that could easily get missed in a busy lab.
This weird growth pattern leads us right into its profile of deception. Because identifying Elizabethkingia meningoseptica is not always a simple task. Thanks to a really tricky biochemical profile and, believe it or not, a very complicated family tree.
So when you look at its biochemicals, it's oxidized and catalase-positive, like a lot of non-fermenters. But the one that really stands out, the one you have to look for, is the strong positive indole reaction. This is absolutely critical. It's a key differentiator that can prevent automated systems from misidentifying it as something else, like a Burkholderia cepacia.
And, this organism has had a bit of an identity crisis over the years. It was discovered way back in 1959 by the amazing Elizabeth King, who named it Flavobacterium. Then in 1994, it became a Chrysobacterium. Finally, in 2005, thanks to 16S rRNA sequencing, it got its own genus, Elizabethkingia. Knowing this history is actually vital if you're trying to make sense of older case reports.
And the plot thickens. It's biochemically almost identical to another major pathogen, Elizabethkingia anopheles, so much so that we now believe many past infections we blamed on Meningoseptica were probably caused by Anopheles. This includes that huge, tragic Midwest outbreak back in 2016. This historical mix-up really complicates our understanding of its true epidemiology.
And this is exactly why our modern identification methods are so important. MALDI-TOF is incredibly fast, but its accuracy completely depends on how good its database is. And 16S sequencing, the very tool that helped define the genus, often can't tell the different species apart. So for a definitive ID, especially when you're dealing with an outbreak, whole genome sequencing is without a doubt the gold standard.
All right, so let's say we've jumped through all those hoops. We have a definitive ID. Now we get to the toughest question of all. How in the world do we treat this thing? This is where the challenge really begins for the entire clinical team.
This slide pretty much says it all.
Elizabethkingia is naturally, intrinsically resistant to almost every single drug we would typically use for a gram-negative infection. This isn't acquired resistance, it's hardwired into its genome. It has 2 different chromosomal metallobeta-lactamases that just chew up carbapenems. And on top of that, it's also inherently resistant to aminoglycosides and colistin. It's a real nightmare.
Now here's a super practical point for anyone reporting susceptibilities. EUCAST has not set any clinical breakpoints for Elizabethkingia. Let that sink in for a second. That means we can't report and isolate as susceptible, intermediate, or resistant. The lab has to provide the raw MIC value, add a comment, and that forces a crucial conversation between the microbiologist and the clinician.
And because it's so complex, there are basically no specific guidelines from the major players. Not from the Dutch SWABE, not from IDSA, not from ESCMID. Treatment across the board relies on good old-fashioned antibiotic stewardship. You see your empirical therapy failing, and you pivot fast to a targeted therapy based on local susceptibility results. And the drugs that often work are ones we don't usually think of for these infections, like fluoroquinolones, trimethoprim sulfamethoxazole, or even minocycline.
Treating children is especially difficult, as you can imagine. These examples pulled from case reports really just highlight how individualized and, frankly, sometimes desperate the therapy has to be. You see a lot of combination therapies, but as you can also see, the outcomes are tragically all over the map.
And this right here points to a major shift in our thinking. Historically, vancomycin was used pretty frequently, but now it's largely discouraged. The reason is simple. The MICs are often just too high, and its ability to get into the central nervous system is poor, which makes it a really unreliable choice for the kind of devastating meningitis this bug causes.
So, given that treatment is such a minefield, what's left? Well, prevention becomes absolutely paramount, and that means we have to focus on where this pathogen loves to live, the hospital environment itself.
An effective infection control strategy has to be an all-out assault on the environment. It kicks off with aggressive surveillance. We're talking systematically culturing water taps, sink drains, even medical equipment. If a source is found, remediation can be as drastic as shock chlorination or even replacing entire plumbing fixtures. You couple that with enhanced disinfection and, of course, strict patient management like contact precautions and cohorting.
And that brings our journey full circle. It leaves us with a final question for you to really think about. In your own institution, in your own lab, what are the biggest challenges you run into when you come face-to-face with a non-fermenter like Elizabethkingia, one that just completely throws your standard protocols out the window?
We would genuinely love to hear your thoughts on this. If this kind of deep dive into complex microbiology is your thing, please subscribe for more. Make sure to leave your feedback and any questions you have in the comment section below, and please share your own experiences with Elizabethkingia. Thanks so much for watching.